Okay, so for 1.2, a more important part compared to 1.1, because for 1.1, we already learns about the viruses. So for these topics, then we look at the cells, okay? So viruses, they are non-cellular structure, non-cellular structure, but now we look at the cellular structures, but particularly we talk about the eukaryotic cells, okay? So what we're going to learn, what we're going to learn for this 1.2, okay, what are the learning outcomes that we need to cover? So first of all, we're going to look at describes and interpret. What we're going to interpret? We're going to interpret the photomicrograph, as well as electron micrograph, as well as the drawing of typical plants and animal cells. Okay, so in your SPM or in your IGCSE, you cover some of the organelles, right? So today, or in these subtopics, we are going to look at more organelles that we are going to cover, as well as some organelles that you guys are familiar with. Okay, for example, nucleus, ribosome, mitochondria, chloroplast, cell wall, plasma membrane. Those are you guys are familiar with. But for AS syllabus, we're going to zoom it into more detail. Okay, so what are the organelles or cellular structures that we need to know or we need to study? So first of all, we're going to look at the first one, nucleus, and the subnuclear structure, which are nuclear envelope as well as the nucleolus. Okay, then we have two types of endoplasmic reticulums. Two types. We have rough endoplasmic reticulum and smooth endoplasmic reticulum. Then followed by the Golgi body, okay, or Golgi apparatus, okay, interchangeable. These two terms you can use. So some books use Golgi complex, and then followed by the lysosome. A ribosome, we have two forms of ribosome, which is ATS in the cytoplasm of the animal and plant cells, or 70S in the chloroplast in the plant cell, or mitochondria in the plants and animal cells. And then we look at the mitochondria, and we need to know that in this mitochondria, we have the small circular DNA, compared to the DNA inside the nucleus, slightly different. Then we're going to look at the centriole together with the microtubules, cilia, chloroplast, large permanent vacuole, and the tonoplast for the plant cell, cell surface membrane or plasma membrane, microvilli, cell wall, as well as the plasmodesmata. And then when we study about mitochondria and the chloroplast, we're going to look at how the ATP can be synthesized through the respiration and photosynthesis process. And then we're going to state that where or how the cell actually use the ATP. So ATP is not energy. We're going to talk about this in detail for ATP. Okay. And then last, for these subtopics, we're going to compare the structure of the typical plant cells compared to the animal cells, which are the structure in terms of the difference in the structures, as well as what are the organelles actually only appear in the plant cell, or what other organelles only can be found inside the animal cells. Okay. So before we study about these cells, okay, we talk about the cells, we need to know a little bit about the cell theory. Who are the one actually give the name of the particular structure called a cell? Okay, so we're going to look at a bit of history, which we're going to look at is in the cell theory. Okay, so now what is means by the cell theory? So when we talk about cell theory, basically means that we refer to the idea that for all basic units, okay, of structure in every living thing, for any basic unit of the structure in every living thing, the ones that have the functions here, or the smaller units here, is known as cell. Are you clear? So if you remove the cell, you zoom into, even smaller structure, you can say, oh, atom is a smaller structure, but atom won't give us any functions in terms of living. They are not living in terms of atom. Are you clear? Look at the atom, carbon atom. You look at the oxygen atom. Definitely, they have their functions, but it's not for living. They are non-living. So as long as we talk about living thing or living organism, our basic unit of the structure is known as the cell. But if you look at the study of the cells compared to other study, for example, classification, for example, the ecosystem study, you realize that the study of the cell is quite recent. It's about 350 years ago. So quite recent, you compare to other kind of study. The reason here is no one knows about the cells before the inventions of the microscope, because the cell is so tiny. Okay, none of us can actually use our naked eyes, okay, naked eyes to look at these cells, right? So you cannot say, oh, this is a cell. How? Because we need to have the microscope. Are you clear? So without a microscope, no one actually can visualize the cells. So with the inventions of the microscope, that is the time people actually, or scientists, study on the cells and come out with the cell theory. And the two most famous and given the credits for the efforts or their contribution in the cell theory, these two persons, they are Germany scientists. One is a Schleiden, another one is a Schwann. Okay, Schleiden and Schwann actually articulate or formulated out this cell theory in 1838 and 1839. It's quite close in terms of both of them. One study in terms of animal cells, another one study in terms of plant cells. They did not know each other. Okay, in the history, they did not know each other, and they present their work. They share their work, okay, their research work, and suddenly realize that both of us, means Schleiden and Schwann, suddenly realize that both of them actually observe onto the same thing known as the cell. So they form three statements here for the cell theory. So what are these three statements? So they say that the cell is a unit of the structure, physiology, and organization in the living things. So means that for any living organism, they must make up of the cells, the structure in terms of the cell. Physiology means function of the cells, and the cell can be organized inside this living thing. Second statement, the cell retain dual existence, means two form. It can be a distinct entity or a building block in the constructions of organism. So what's mean that distinct entity? In this case, you refer to the bacteria, refer to the amoeba, where they are unicellular right now. So what I mean unicellular? So it means that if you remove all the cells, leave them alone, they can still survive. Okay, they do not need to depend on other cells. So unicellular, or the building block in the construction of organism, means that they can be multicellular. Are you clear? So what's multicellular? So basically means that the cells, okay, this is the kidney cell involved in the excretion. Oh, this is a liver cell involved in the detoxification. Oh, this is a lung cell involved in the gas exchange. So it means that they're built up. So it means that something like a Lego, something like a Lego that we can actually put them together, make it a complete one. If you use a Lego block, a square one, seems like no functions. Can you see that? But when you mix and match all together, it forms a complete block. So this complete block is known as a multicellular organism. So this is a second statement. And the third statement come out with cells formed by free cell formation, similar to the formation of the crystal, known as spontaneous generation. So this is something fun. If you look at it, what's with spontaneous generation? So there are two scientists actually observe some phenomenal, okay, happens in their surrounding. One of them actually look at this, the meat. Again, they look at the meat. So they put the meat on the table. On the table, they observe. One day, second day, third day, then they realize that maggot come out from the meat. Okay, you know the maggot, the larvae, right? The maggot come out from the meat. So when the maggot come out from the meat, then scientists say that, oh, this is how the life is formed. Okay, out of nothing, up, generation, spontaneous generation. So when spontaneous generation, out of nowhere, suddenly pop, living thing come out. Okay, so this is means by spontaneous generation. So at that moment, a lot of scientists want to know how the living things start on our Earth. All scientists actually want to know, okay? They're so surprised. You look at the moon, you look at other planets, no living thing, so-called no living thing, but why there is, or there are living things or living organism in our on our Earth. So how this living organism actually coming from? So scientists actually think when they look at this meat and suddenly got maggot come out, then they know that, oh, spontaneous generation take place out of nowhere. Okay, it may be like this. You look at the Chinese movie or the drama we call Journey to the West, where the Su Kong writes out of nothing, then pop out from the stone. So this is something known as spontaneous generation. Out of nothing from the air, suddenly the living thing come out. So this is first observation. The second observation by another scientist, basically at the dustbin. So they throw, that means the thing, okay? They throw the rubbish into the dustbin, okay? So in the dustbin, only the waste. But the second day, when the scientists opened the bin and they saw the rat inside, saw the mouse inside. So they thought, oh, out of nothing, the rat can come up like this. So they also term it as a spontaneous generation. So now, a quick one, answer the question here. Out of these three statements, which one or more than one statement is no longer valid today? I mean, with the more advanced technology, we know that, oh, this statement, whatever the statement articulated in 18th centuries, now may not be valid anymore. So put a tick, okay, put a tick if the cell story, this cell theory is still valid. Put a cross if the cell theory no longer valid. Okay, so now answer in the check box here. If you think that the first theory is yes, still valid, put a yes there. The first statement. Still valid or not? The first statement. Yes, I saw lots. Yes, yes. Okay, yes. Okay, so look at the answer now. The first one, actually, yes, still valid. Means that the cell is still the basic functional unit of the organism. Okay, without the cell, basically means that no living organism. How about the second one? Yes or no? In the chat box now, the Zoom chat box there. Yes or no? Okay, good. A lot of you guys put a yes, so let us review the answer. Yes, it's still valid until now. Are you clear? Until now, 10 years, 20 years after that, you may not know. But at this moment, yes. Okay. So cells, the third statement, cell formed by free cell formation, similar to the formation of a crystal, known as spontaneous generation. Yes or no? Yeah. Yes or no? Okay, good. So let us look at the answer now. The answer is no, because we do know that living thing cannot, out of nothing, pop, out of nowhere, they come out something. Okay, then it's very, very creepy. You try to imagine that your pencil suddenly pop, from pencil it become living organism. Okay, so out of nothing, then the cockroach come out. So this is called spontaneous generation. It's very, very creepy, and you're gonna freak out. Okay, if let's say this is real. Okay, so at that moment, now we know that spontaneous generation is not valid. Means that living thing cannot just pop up from nothing, from the air, then pop, living thing exists in this case. So who actually reject this statement? So the one that reject this statement is known as the Rudolf Virchow. Okay, in this figure A here, 1.5A, this is the picture of Virchow. So what Virchow actually said, Virchow actually added the third tenet of statement to the cell theory to complete the entire cell theory here, with the Latin tenet stated omnis cellula e cellulose. With the translation means all cells arise only from pre-existing cells. This is the statement. So means that the living organism won't form from nothing. It must from the pre-existing cell, which is by mitosis in the asexual reproduction, or by meiosis when the fusion of the gamete take place. Correct or not? So means that whatever cells we have, we must inherit it from the pre-existing cells. Now everything go back to the square. Why does it say go back to the square? Because initially the scientists think that, oh yeah, out of nothing, this is how the life actually come out or emerge, right? So it means that on this earth, actually nothing, no living organism. One day, suddenly, cells come up. Oh, the thing, pop, come out. Correct not? But now we know that all cells must arise only from the pre-existing cells. So what's this mean? We go back to the square. How the first living organism actually arise on our earth. So until now, scientists actually don't have a very, very concrete evidence to say that because of what events actually the living organisms start to form on our earth. Are you clear? Okay, no one knows until this moment, but most of the biologists want to know how the cells actually or living things start to be formed in our living on our earth. Okay, so once these standards added, the classical cell theory now complete. So what are the three theories here? So first, the cell is a unit of structural physiology and organization in the living things. This is correct. Second, the cell retain a dual existence as either unicellular or multicellular. And the third tenet here is called all cells arise only from pre-existing cells. Okay, so these are the three cell theory, or we call classical cell theory. Okay, so however, the idea of all cell called pre-existing cells has in fact already been proposed by Robert Remarque. So number B here, Robert Remarque actually formed this, but the one that make it famous, okay, is Virchow. Virchow actually make this tenet, okay, become famous. Okay, so just a quick history for you guys. But no matter how, when talk about this cell theory, we need to pay the or to give the credit to these four people here for their contributions onto the inventions of the microscope. Because no matter Virchow or Schleiden, Schwann, they won't be able to study the cell theory if there is no microscope. So basically, the credit should be given to these four people. So we have the Hans and Zachariah Janssens. Okay, is not mistaken, is the father and son. Both of them actually, they are the father of the compound microscope. What's the father of compound microscope? They are the one that invents the compound microscope, not simple microscope. So what is the difference between compound microscope and the simple microscope? Compound microscope consists of two lenses. Okay, two lenses. One lens, when we view it near to our eye, called as the eyepiece. Another one lens near to the object is called objective lens. So Hans and Zachariah Janssens are the ones that they were the ones that invent this compound microscope. Leeuwenhoek is the person that actually have a skill to polish the glass, okay, the lens. When they polish the lens, they make the higher magnification so that you can view more things. So Leeuwenhoek. And Robert Hooke is the one that actually the first person described the cell as a cell. Okay, so with these three a brief idea of their contribution. First, Hans and Zachariah Janssen, both of them invented the compound microscope, not simple microscope. Simple microscope means consists of one lens only. But Zachariah and Janssen invented the microscope with two lenses. One, we can view it near to our eye, is known as the eyepiece. Another one far away, means from our eyes, but near to the objects, which is known as the objective lens. Okay, so second, Robert Hooke is the one that viewed the cells and then describe that, use the term as a cells. Are you clear? Okay, and Antons van Leeuwenhoek was the ones that actually improve the lenses. okay, improve the lens, and then make the this microscope have higher, so-called microscope, the handheld microscope, to make it more have higher power of magnifications, okay, so that they can view even more type of cells. Okay. So now I have this contribution and a scientist. So can you fill it out for the first contributions? Who are them? So credit to the first compound microscope, usually give to him, actually is to them. So one lens was at the eyepiece of the microscope, we call ocular lens. And second lens near to the sample of study, known as the objective lens. Okay, ocular or eyepiece, another one objective. So this one contributed by who? Zachariah, Hans and Zachariah Johnson. Okay. So the second contributions here, English scientists who discovered the cells in a piece of cork. It's a cork cells. Sorry, it's a cork cells, which examined under his primitive microscope. So look at this primitive microscope. It's the model of the microscope that we use it currently. So this, you can see they're very, very primitive. So you can see that what actually happened here, how to use it. You can see that this is the part where you put in, okay, we can put our specimen, it's a specimen holder. And then we have the lens here. So this lens near to the object that we want to view, so it's known as the objective lens. And then the one that we put our eyes, okay, here to view is known as eyepiece. Similar to our microscope, right? Can you see that? Eyepiece and the objective with the specimen holder. So in order to have the light and focus the light, they use the water flask. Because water flask, you know that, okay, right? It's a convex, convex surface, and it can focus the light onto our specimen. So where is the light source? They use the oil lamps here, so the light will be focused onto the specimen and view. So this is a very, very primitive microscope used by who? By Robert Hooke. So Robert Hooke actually going to draw out the structure. This is the structure. Can you see that? In this structure, you can see something like a room, a lot of room, right? So because the word cell, C-E-L-L, basically means that the room or the prison, right? The cell, C-E-L-L, the prison. So Robert Hooke look at this structure, it's something like a prison, the room or the prison, and then he termed this structure as the cell. Are you clear? Okay. But Robert Hooke at that time, he didn't know that what he observed actually was or were the dead cells. Are you clear? What he observed basically was the cell wall of the cell, because when the cell died already, the cell wall remained. The cytoplasm all degraded. So the remaining, what he observed actually is not really a cell, it's not a complete cell. What he observed actually was the cell wall, the remaining of the cell and the plant cells. Are you clear? And he termed it as the cells. Because why? It looked like the prison or the room. So therefore, Robert Hooke actually gives a name, okay, called the cells. So the word cell is derived from Latin word cellula, which means small compartments or small room. Okay. So the contribution here is by Robert Hooke, okay, by Robert Hooke. So therefore, the word cell, we use it until now. I try to imagine if the Robert Hooke looked at different type of the cell and saw the star shape, now maybe we call the cell as a star. Cannot. It's just because of Robert Hooke called as a cells, now we use it until now. Okay. So what he observed actually is not really a cell. He observed actually is the cell wall of the plant cells. Okay, no cytoplasm degraded. Okay, so now the last one, you know, that is the Anton van Leeuwenhoek. So what he did, okay, he used his single lens monocular microscope. It's something like this. Can you see that? So how do you use it? You can see that this is where you can place the sample and the lens. Can you see that? The lens. So he just need to view it under the light. So from the light will focus on this specimen and he can enlarge. Why? Because he has the skill to polish or to make the lens, together with a naturally acute eyesight and great care in adjusting the lighting where he worked, he managed to magnify over 200 times with clearer and brighter image. Okay, so that now he can actually view the blood cells and also the protozoa. So means that from his work, because we know that plants are larger, are you clear? Plants are larger, we still can use a what we call this lower magnifications microscope to view the plant cell. But to view the animal cell, you need to have higher magnification. So because of his work, now we will be able to view the protozoa or the blood cells at that particular era. Okay, so by Anton van Leeuwenhoek. Okay, so with the advancement in the technology, okay, now we do have a very, very sophisticated devices to help us to view the cells and study the biochemistry of the cells. Now we do add in more and more cell theory, okay, and form this what we call the modern cell theory. So what are the modern cell theory? So modern cell theory still consists of that particular three classical cell theory. On top of this classical cell theory, we add in another four. So what is the first one? So the first one, actually, the cell is the fundamental unit of the structure and the function in living organism. Means that if you look at the organelle alone, it's not a functional unit, because organelle still need to depend on the whole cells. Are you clear? And number two, all known living things are made up of one or more cells. So it means that either unicellular or multicellular. Number three, all cells must arise from the pre-existing cells by divisions. So it can be either mitosis or meiosis. Okay. And number four, you must have the energy flow occur within the cell. So what's mean energy flow? So it means that we have the biochemical reactions. You need to have the input of the energy. So that's why we need to eat. Can I see that we need, the plant need to carry out photosynthesis, correct or not? So that is how we carry out respirations to have our own ATP, okay? So next, cell contain the hereditary information, the DNA, thanks to these two persons who are Watson and Crick. Watson and Crick were two persons that discovered the structure or formed the structure of the DNA. And we do know that this DNA is the hereditary information that pass from one cell to another cell during the cell divisions. And number six, and we know that all cells are basically have the same chemical composition in the organism of a similar species. So it means that that's why we know that similar species are human, tiger, okay, because they consist of almost the same chemical compositions. And the last one, the activity of organism depend on the total activity of independent cells. So what does this mean? Neurons work itself, muscle work itself, but the overall effect, you must depend on both of them. Are you clear? So for example, our sensory, our eye. Okay, for example, you saw someone, okay, you see someone on the hit on, slap you. What you did? Your sensory neurons that function, right? Your eyes interpret the signal sent to your brain. So they are independent. Can you see that? The eye cells work itself, brain work itself, muscle work itself, right? But the total overall effect actually depends on the total activity of these independent cells. So these are the seven. Okay, these are the seven statements formed in this modern cell theory. Okay, so what we're going to study them in your AS and A2 syllabus. Basically, when talk about the cell is the fundamental unit of structure and function in living organism. This will link with our chapter number 1, where we're going to learn about the organelles, how to make sure that the cells is a functional unit. So the cell that without the organelles, the cell also can't function. So that's why first chapter we're going to learn. Okay, so all known living things are made up of one or more cells. So therefore, we can see that forming different kind of the multicellular organism or unicellular organism. So we need to do a classification. Right? We need to classify that, oh, this is human. Oh, this is tiger. Oh, this is bacteria. Oh, this is a plant. So we're going to learn it in A2. We talk about species and also the biodiversity. okay, in A2. The next, all cells arise from pre-existing cells by divisions. We're going to look at in mitosis in AS, meiosis in A2 syllabus. Number 4, energy flow, metabolism, and biochemistry. So we look at the enzyme, how the enzyme functions occur within the cells, how the cells carry out the aerobic and anaerobic respiration, how the plant cell carry out the photosynthesis, involve the energy flow. So AS and A2 process syllabus again. Cells contain hereditary information which pass from cell to cell during the cell division. Again, we're going to learn this one in the DNA replication. So how your DNA replicate before they can divide. Okay, this DNA can be divided into 2 cells. Okay, and also basically the same in the chemical composition in an organism or a similar species. Again, how we actually interpret, oh, they are same species, or why humans and monkey 2 different species. Can I see that? Okay, how the dog and the wolf, some of them belong to the same species. We're going to learn this one in the selections and evolutions. And the activity of organism depend on total activity of independent cells. We're going to look at the communication between the cells. Particularly, we talk about the homeostasis as well as the muscle contractions. Okay,